effort. Next week is our absolutely final seminar of the semester, and the week after that is our TESS retreat. So the TESS retreats, if you haven't talked to anybody, everybody has a really good time there. I don't know if it's still possible to sign up, but a couple of days, and I think it's a way to really get a good picture of all the research that's happening around you here at JCU, as well as lots of other stuff, free alcohol, et cetera, et cetera. So anyway, we'll go ahead and pleasure to introduce Professor Lynn Schwarzkopf. I guess you have a personal chair at JCU. Lynn is a Canadian national originally, of course now half Aussie. She did her studies at the University of Guelph for her undergraduate and her master's degree, and she came to the University of Sydney to work with the big grand frogman, Rick Shine, Professor Rick Shine, who's one of the most eminent and highly cited scientists in all of Australia. After that, she had some postdocs. She went back to the University of British Columbia in Canada, a couple postdocs here, and she's subsequently had a position here at JCU for 35 years. So she knows where all the bodies are buried. And watch your steps, because you might just tell. Anyway, thanks very much, Lynn. A pleasure to hear from you today.
Okay, thanks very much. Thanks very much for the invitation to come to Cairns campus and have a look around. It's always good. Remember where everything is in Cairns and then leave for long enough to forget all about it again. And today I'm going to be talking to you about some of the work we've been doing in our lab with a big group of people working with ecoacoustic data. And what I'm hoping is that some of the people listening have ecoacoustic data they don't know what to do with, and hopefully I can help you, but maybe not very much. So I'll just start by acknowledging the traditional custodians of the land and pay my respect to elders past, present, and emerging. Okay, so what is ecoacoustics? Ecoacoustics, most people probably know, they use acoustic recording to obtain information about ecosystems. And it looks at sounds and acoustic patterns produced by organisms, whatever organisms, in their natural environment, and investigates the influence of ecological processes and also environmental processes on those organisms. So we can hear some environmental processes and we can measure some other environmental processes at the same time as recording. And so we can actually really look at ecoacoustics. And the recent development of really small acoustic recorders and with long battery lives has really changed the whole scene here, so that now we can record for a really, really long time in the field. And that activity, sticking a recorder on a tree, is called passive acoustic monitoring, or PAM, and I will use that throughout the talk, maybe sometimes. So these are the things that you may or may not be familiar with from your own research. So up in the corner there's an audio moth, little tiny, very inexpensive recorders. The next thing beside it there is a solar bar. Oh, I can probably point to it here. Yes, so that's a solar bar. That's a solar thing, and it lasts the entire year, and it can record for a whole year. It's got all kinds of SD cards in there. This is a song meter. This is a thing called a bug that they're using in Borneo. This is another one of the bars. So this is a bar LT, which is a long-term bioacoustic recorder built in Australia. That's the insides of one. And that's a hydrophone for any marine people who might be listening. Okay, so there's lots of advantages of PAM in relation to those recorders that I just showed you a picture of. It's not a lot of initial effort. You know, you go out with your little recorder and you stick it in a tree, or you stick it on a pole or something like that, and then there it is. So you put them out and you leave them there for some period of time. So that's really easy. Even computer scientists can do it, which may or may not be an advantage. It records when you're there, and that eliminates interference. And it's something that I hadn't really thought about, but the thing's recording when you're not there. And we don't know what happens when we're not there, do we? So this is kind of cool, kind of Schrodinger's cat kind of thing. And it allows permanent data storage. So we can save this data into the future, which is going to be necessary because we don't know how to analyze it. But anyway, we are saving this data into the future, and it could be useful. And experts do not have to be present during survey, which is convenient because sometimes it's really hard to get enough birdos. We're always struggling to get enough birdos to come out in the field to recognize the different calls of different things. And then if you're doing a lot of things, you need not only a birdo, but you need a froggo, and you need all these different people. So it makes it easier to collect this stuff and then get those guys. And then they don't even have to get sweaty or get bitten by things. So it's easy enough. You just record things in the field. Sorry, I'm not supposed to walk around. You record things in the field. That's a hydrophone, but you get the general idea. You can store this stuff. Then you magically do your signal analysis. You look at it in relation to environmental things, and you get a lovely idea about what's happening with ecology. The disadvantages of PAM, of passive acoustic monitoring, is that you get a lot of data, like a lot of data. And analysis is a problem. So right now, we have no problem at all with this part. We can put something on a tree and record. We have no problem at all with this part-ish, like we generally can store the data that we've collected. But then you see it all gets very foggy and we don't know really what's going on in there. And you see the smooth sailing then turns very, very gnarly. And it's just difficult to analyze the data once you have it. And that has been a problem in general with ecoacoustics. Mainly, passive acoustic monitoring has been used to look at single or a few species. And actually, a lot of active acoustic monitoring where people are sticking a microphone in something's face are what people are doing with recording. That happens a lot. And so they may record behavioral interactions and things like that. They might look at what happens at nests. They might look at what happens when there's an interaction like fighting, when things are feeding. They might collect entire behavioral call repertoires for different individual species. They might detect the presence of breeding behavior, for example. If you're trying to do many species, and ecoacoustics is kind of aiming anyway to do many species, then you might detect the presence of something because you hear it call. You might detect or describe the activity of that animal. And sometimes, often now, lots of people have done studies on this now, activity tends to be related to abundance. It's not that shocking, but it's nice to know. You can examine vocal activity in relation to the environmental things, which is what I was talking to you about before. In general, passive acoustic monitoring has worked really well for bats and for marine mammals. So those two things have been studied quite extensively, and there's quite a lot of tools for studying those guys. And then there's a few animals, Australian things that people have worked on. Plains wanderers. Every time I go to an ecoacoustics conferences, about 50% of the talks are on plains wanderers. They're this little bird here. Glossy black cockatoos are well studied. Kuranda tree frog is well studied by Conrad at JCU. And koalas are really well studied in New South Wales. But it tends to be these little individual animals that are studied. And so this comes from a paper by a lady called Larissa Sugai, and it just shows how people analyze their data from these studies, from these kinds of studies. And what you see is that, I'm not allowed to walk around. What you see is that most of the studies that people do involve manual listening to the data. That's how they analyze the data. So there is a bit of automatic stuff. There's some semi-automatic stuff. And people use indices, which I'll talk to you about later. But this here, manual, is still, this study was done in 2019. People are still listening. That's the main way that they study acoustic stuff. And this just shows you for individual groups. Here's bats, non-flying mammals. There's hardly any studies on non-flying mammals. Birds, anurans, invertebrates, all these things. The dark blue here is manual listening to the recordings. So mostly people study these things by manually listening to recordings. The only people who don't are people who look at soundscapes, and that's because you can't manually do a soundscape. So I'll talk to you a little bit about soundscapes, but not for very long. So you can monitor the entire soundscape. So one thing that these recordings do give you is all this complex noise. And you have a lot of noise, and you have a lot of it over a long time. And an approach that's been taken, particularly by the Europeans, is to do soundscape monitoring. And so this shows you, this is a graph, and this shows you an index. And the index is called soundscape saturation, which is an index divided by a different index. And it shows, it just is sort of how noisy is it, is sort of a measure of that. The orange line is a logging concession. The blue line is a different logging concession. And the green bit is protected forest. And this is for a rainforest. And it shows you that over time, there looks like there's differences among the different indices, among this index anyway, in this index. So, you know, the nights appear to be quieter, because this is a day I'm along here. The nights appear to be quieter in the undisturbed forest. And then there's some daytime noise. and it looks like maybe this part of the morning is noisier in the protected forest, but maybe the dawn chorus is louder in the concessions. Maybe some other parts of the day, the night is definitely louder in the concessions. And so the thing that I find unsatisfying about this is that while you can definitely get patterns, and you can definitely see the impacts of things on indices, whatever index you want to look at, there's all different ones. This happens to be percent saturation. There's also the acoustic complexity index. There's also the biodiversity index. There's also event. There's also entropy. There's heaps of them, probably 30 or 40 different indices. We cannot yet, in my experience, predict what the indices are going to do in response to anything. We can look afterwards and say, oh, look, the H index is higher, or the percent saturation is lower between 2 o'clock and 3 o'clock in the afternoon. And some people even use those and kind of say, okay, there we know the impact. But I think we don't really understand what we're looking at yet with indices, which is kind of sad but true. So basically, the best use of these soundscape analyses is not very clear to me anyway at the moment, because we cannot predict ahead of time what we should see if there's a bad impact on an ecosystem or a good impact on an ecosystem. Instead, it's always done post hoc. We can distinguish habitats and impacts for sure, but they are not, it's always post hoc. They're not predictive. And they keep picking new ones when it doesn't work the way they think it's going to. So indices tend to vary for really unclear reasons, and we're working on indices too, but we have not got very far with them. They need a lot more work for community ecology. It cannot be used for community inventory in any kind of normal way. So we thought, oh, this is easy. We just look at acoustic complexity. When it's really high, it means there's a lot of animals. When it's really low, there isn't so many animals. No, that is not true. As a matter of fact, quite the reverse sometimes, and I don't know why. So one thing is that I want to point out here, and I'll have a reason for pointing it out in a minute, is that very few studies do inventories. So this is just looking at the number of articles that do inventories, that talk about the species of things. So there's a few studies that have looked at how many species of invertebrates there are. So this is unrelated to indices now. This is where people are actually counting the species in the data, okay? And so there's a few on invertebrates, a few on non-flying mammals. Soundscapes are always indices, so it's not that. Frogs, there's a few. Birds, there's a few. And bats are the most common group where they actually do the numbers of species in the sound that they're listening to. Unfortunately for the bat people is that there has been a bit of a discovery recently that what they thought were species-specific calls are not. They're activity-specific calls. So when bats are feeding, they make some noises. When bats are echolocating, flying through a forest, they make different noises. But the different species of bats make the same kind of noise. And so their species inventories are maybe not as reliable as they thought. They still can absolutely get species differences, but it maybe is more fraught than they thought it was originally. So the question we wanted to solve in my lab is, can we actually use passive acoustic monitoring for species inventories? Is it going to be possible to do that? Because I'm not very satisfied with those indices. I want to be able to get the species out. That's what I want to know. What species are there? So this all started with the Australian Acoustic Observatory. So the Australian Acoustic Observatory is a bunch of sites, 86 sites. Apologies to people who've already heard about this a zillion times. 86 sites with 4 recorders at each site. The recorders record 24/7 for 5 years. They record for a full year. They can record for a full year before anyone needs to change the SD cards. They are not networked. We go out and change the SD cards for various reasons, partly because Australia is not very particularly well networked. That is a lot of hours, which adds up to a lot of years. 1,994 years of sound in 5 years we will collect, which is stressful when you think about it that way. But don't think about it. So every single site has this particular design, only it doesn't look as neat as this usually. So it's got 2 sites that are established near wet. So these things are supposed to be the bars, the recorders that I showed you, the solar bars, a box with a solar panel on top. So that's what this is. We got 2 that are close to within 50 meters of a water ish body because we're in Australia, so a lot of times they don't have any water in them, but you get the idea, water course of some kind. And then 2 of them are further away from water, at least 1 kilometer away from water, or at least 500 meters away from water. These things are 500 meters apart, just so we don't get the same animal on the recorders. And like I said, they don't look anywhere near as neat as that, but that's the general idea. And so at every one of those sites, there's 4 recorders. Okay, so we did that. And then we thought we would need to ground truth what was actually happening at some of the sites so that we could compare it to the audio, so that we can understand what was happening in the audio and know that we knew what was, it relates in some way to the real world. Because if we're going to use it for monitoring, and especially if it's going to replace some of the monitoring that we do by going out in the field with experts, we really need it to work. It needs to do that properly. So we monitor amphibians, reptiles, birds, and mammals, and we do that with manual or traditional surveys, as well as with audio monitoring. Okay, and so we've got our sites. These are our 6 sites that we've selected where we do our actual physical monitoring. And it is also, of course, sites where recording is happening. So that's happening there. And we went to these sites twice over 2 years in theory, except there was COVID, and then there was floods, and then there was pestilence and everything else. But we tried really hard to get to the sites twice over 2 years. Some of them were only visited 3 times. But we did try. Oh, there was a plague. There was actually pestilence. So we actually had a plague of mice in Queensland at this time. Anyway, that's a long story. So the 2 people who are responsible for this in the main are my postdoc Slade Ellen Ankins, who is an unbelievable person and does everything, and Seb, who worked his butt off collecting data at all these different sites, actually physically collecting data. And so he went out and did manual or traditional surveys with all the usual kinds of things that you might expect. So there were pit traps, there were funnel traps, there were cage traps, there were camera traps, there were those things, Elliott traps, and these things called cover boards, which no one's ever heard of, but they're a reptile thing. And so you attach these things to trees, and then when you take them off, there's geckos and little things hiding underneath there. And it's very convenient because they're hard to get any other way. And that way you don't destroy the habitat, because we were going to be returning to these habitats again and again. And the way you do it, if you're a normal herpetologist, is you rip all the bark off the trees. And it's not very nice for the geckos because they can't use those trees again. So we use cover boards. And we also do bird watching, regular bird watching at the sites. And we also go spotlighting at night at all the sites. And they go to a site for a full 7 days, and they record all that data while they're there. Meanwhile, the recorders are going, which is humorous when you hear them talking to each other on the record. Anyway, and so the idea is that we want to compare the manual results with the audio results. That was the plan. That was my brilliant idea about what we were going to do when I applied for the funding. So we have a bit of a problem with this, because at our sites, there's at least 407 vertebrate species. And actually, I don't know if this is true. I found this somewhere on the web, you know how it is. They claim that Eastern Australia is the world's newest biodiversity hotspot. And if that's true, we made a serious mistake. There's so many things here. Anyway, 280 of these things are vocal, and it's about 221 birds, 31 amphibians, about 25 mammals, and 3 reptiles. And so that's a lot of animals that are at all these sites, that were a lot of different species. And so the thing everyone always asks me when I say, okay, and then we needed to analyze the audio to be able to compare it to our manual surveys, right? And everyone says, isn't there an app that does that? And it doesn't really exactly work for Australian birds yet. It does for some Australian birds, and we'll return to this later. But it also doesn't do mammals or amphibians. You'll notice there's pictures of birds on Merlin. There's a picture of a bird on BirdNet. BirdNet is also called BirdNet, which is a bit of a hint, okay? So we still needed to do some stuff. And so hence we entered our journey, okay? And so I had no idea when I started this that no one uses audio for species inventory. It just seemed completely obvious that you would, you're going to hear all these things and they're going to be on the recording and you'll be able to measure them and it'll be fantastic. That was what I thought. Little did I know. So here we are at the beginning of the hero's journey. Okay, we've got all our known stuff, which is the manual surveys, but now we have to analyze the acoustic data. Okay, and so the first thing we did is we ran into, I don't know if you can see that, but we ran into the threshold guardians. Okay, and the threshold guardians were computer scientists, and they said you have to build CNNs. You have to build CNNs. And CNNs are convoluted neural networks, and they're a way of analyzing sound. Okay, so computer scientists do pictures like this, so apologies. But anyway, what you do is you make a time frequency spectrogram, which is this thing here, of sound, and I hope you can see it. It's purple. The red is the actual sound that you're interested in. Signals are detected within the recording, these boxes here, and they're labeled. Okay, so somebody sits there and puts a box around them and labels them, says what they are. And then you hand design a classifier that can find the characteristics of that sound. So it describes it according to duration, maximum frequency, minimum frequency, peak frequency, harmonics, all kinds of things where you can describe that. And you can do that by hand. And if you can either describe it by hand if you like, or you can learn it from the data structure, which is called deep learning. So you can use fewer examples labeled by somebody, and you can learn it from deep learning. And then that gives you, what happens is once you run that CNN over your data, it outputs all your, it outputs the hits. It tells you where that species is called. Okay. The problem with it that we discovered was that you need labeled data, and they're not joking when they say you need labeled data. So it's not like you need 100 labeled examples or 1,000 labeled examples. So if you think about Google says it can tell you, like when you look up cat, it can show you a picture of a cat. That's because hundreds and hundreds of thousands of people put up pictures of their cat, labeled cat. And so they've got all this labeled data already. And so having labeled data is incredibly important, and you can build a CNN literally for anything as long as you have enough labeled data. And so at first we thought, oh, as long as you have enough labeled data, no problem, we'll just label a bunch of data, no problem. We had no idea how big, remember how many species we're talking about, over 200 species. We would have to label hundreds of thousands, or at least thousands and thousands of examples for each one of those species. So it's a lot of work per species, so that was a bit of a problem. Also, you never find anything you're not looking for, but we gave up on that ages ago. So we just don't even try anymore. And also, it's not a big problem. So what we needed was a helper, right? And that's what gets us over this threshold, is if we have a helper or a mentor, but mostly helper. So what we thought we would do is we would get labeled data via citizen science. And so we have a couple of citizen science platforms. These are kind of terrible pictures, especially in the light. But we have a thing called Frog Song, and we have another thing called Bird Song, and we can put up recordings, little snippets of things, and our lovely, lovely volunteers, who are faceless people all over the world, identify them for us, and they say whether or not they are actually a particular call. They say, yes, it's a this call, or no, it's not a this call. And so that's really, really nice of them. And so we have our helpers, and they help us do that. What was I going to say about it? Yeah, so I think the citizen science is certainly a way to get labeled data. Oh, I know what I was going to tell you, because you'll all worry, what if they label it wrong? So it's okay if they label it wrong, because we put up every single clip 8 times. And so 8 different individuals, and the same individual doesn't get that again. Only one individual gets it. So 8 people identify that call. If 7 out of 8 people say it's a something, we think, oh yeah, cool, it's a something. If 4 out of 8 people say it's a something, we say it's not identifiable, and we get rid of it, especially if we get a lot of variation. We can also, for things like that, where let's say we get 2 different IDs on that clip, we then give it to an expert, and the expert says, no, really, it's a whatever. Okay, so that was great. So we had citizen science, and that was fantastic. But then we ran into challenges with citizen science. And so here we are in our journey. See, we've run into challenges with our helper. And so what happens is citizen science requires a lot of care and maintenance, okay? Preparing data for the platforms was very time consuming, and preparing tutorials for the platform so people can go back and listen so that they learn bird calls and they learn frog calls took a really long time. The tasks need to be moderately challenging. If they're too easy, if you just ask someone to do a single species, they get really bored really quickly. This is a bored person doing citizen science here. And so they move on. They don't do our job anymore, which is sad. They need to have occasional success. So you can't just put up all the no hits from some other way of classifying things, because then all they get is they never get the thing they're looking for, and they get really sad and they stop doing it. So in order to get citizen science to work, it's important that someone is maintaining these sites with lots and lots of really rewarding clips, with lots of training for different activities, all this kind of stuff. It's great, it's really good, but Slade hates it. So we're not doing it anymore. Well, we are, but right now, everything on our citizen science platforms is identified. And he's got to put another load of stuff up there, and he's not happy about it. Okay, so then we had a revelation. Okay, and that's what has to happen after you face your challenges, you have a revelation. So our revelation was clusters. We thought, oh, this is what we'll do, we'll cluster the data. And there's a program called Kaleidoscope, which clusters data, and it puts it all into different clusters. And the idea is that every single cluster is a different kind of bird. You manually listen to 10 calls in each cluster and you label it, and it's going to be a different kind of bird or a different kind of mammal or a different kind of whatever. Okay, and so we thought, okay, that's really good. And for this, it turns out we needed a helper. So our helper has been my student Thomas Napier. He's actually working on clustering the data for us. We're not using Kaleidoscope anymore, because when we used Kaleidoscope, we found that while it did successfully cluster sounds, what it does is it chops all the audio. All clustering does is they chop the audio up into 4-second chunks, or it's actually 3 seconds, I think. And 3 seconds of data might have a magpie and a honeyeater in it. So you get a cluster that's magpies, a cluster that's honeyeaters, and a cluster that's magpies and honeyeaters. And then you get, and imagine that for every pair of 200 species. So it's a problem. So it reduced our data. It did reduce our data from many, many thousands of sounds to 4,000 to 7,000 clusters. 4,000 to 7,000 clusters. We had to listen to 10 in each one of those. Okay. So each combination of sounds was a cluster, and that was our problem. It was too labor intensive for us. We actually ditched the clusters. However, we now have Thomas, and Thomas is working on making clusters more useful for us. So he's using AI to split up the data first and then identify the clusters for us. So we've got our computer scientist, we've got our helper, and he's doing that. We could have constructed recognizers for these things, but basically the process was just not quick enough. It was just still too much data, still too much to do. So Thomas is still working on clustering, so we haven't left it alone, but we left clustering alone. And then we had another revelation. It was a transforming revelation, and many people here have heard about this, and I apologize. It was a big revelation. So we found this thing called binary point matching. And it's a little R program called Monitor, and you can download this paper now. And what it does is, this is a spectrogram. So this is time on the bottom and frequency on the side, and that's a little call from a bird, okay? And what it does is it puts something over that, and a sort of a mask over the call, and it figures out where there's a lot of sound energy and where there isn't so much sound energy. So the dark places are where there's a lot, and the light places are where there's not so much, and it makes a template. And it says, I will look for places where there is high sound energy in these sort of areas, and there is not very much sound energy in these other areas. So what it does is it passes over the data and it searches for matches with this little template. That's what it does. So it's very simple. It's not even AI. It is automated though. And so this is how it's supposed to work, okay? So let us look first at the blue line. So you see the blue line here, and you see these little boxes here. And these little boxes say, I found a hit. I found a thing that looks like that call, okay? And it found a thing that looks like that call. And when it does find a thing that looks like that call, you get a very high score for that hit. It gives you scores for everything else too. You see this, you get scores, but the scores are all low. This is the theory. The scores are all low, right? But then when it says, oh, look, here's another one. This is a very high score. That's another one of our little calls. And then here's another high score. And that's another little one of our calls, okay? And so the idea is that you can find a threshold, some kind of line like here, which we call a cutoff, which will, above which it's always really the call, and below which it isn't the call. Therein lies the rub, but we won't go there yet. Okay, so let's do the red one. So here's the red box. The red has found some calls here, and it says, oh, look, there is a red one of these calls. And that is the only red one of these calls in this data. Everything else is too low to be scored as a hit using this cutoff, okay? And then you can do the same thing with the sort of mustard colored line. You can do the same thing with the green line. So I think everyone understands. It's really simple. I love it for that reason. You can understand it. And so what happens then is we output, Slade outputs, when I say we, it's always Royal. Slade outputs the top 50 hits for the magpie lark, for example, at Tarcutta Hills. This is a magpie lark call, which is good because that's what we're looking for. So it's saying, here is our week of data, and these are the top 7 hits for every single day of that recording. And what you can see here is that those are all magpies. These are all magpies. These are all magpies. These are all magpies. These are all magpies. That one isn't a magpie. That one isn't a magpie, neither is that one. But it's doing okay. Like in the top 50 hits, an awful lot of them are actually magpies. And we do that. We do exactly that. We check those hits and we make sure that they really are magpies. And we decide on a cutoff after which they're not magpies anymore, okay? So what we do is we build and we run templates of this nature for each species. Some species require a couple of templates, and the templates sometimes require some engineering. Templates don't like it if they don't have much room before them and after them. They sometimes get too much sound, so we need a bit more negative space before and after them, things like that. And some species require template refinement. You might add a little bit more positive space or more negative space, whatever. The advantages of binary point matching is that it's an R program. We can teach volunteers and even undergraduate students can do it. We can run the template fairly quickly, and therein lies another story. We can check the output really quickly. You can look over those top 50 hits in like 2 seconds and see whether you've got hits or not. We can run the templates over all the sites. So we run the templates over all the acoustic data for all the sites. So we can look for things that aren't there, in the sense that if we don't expect to see a magpie lark, we can still look and see if there's any evidence that they're there. And we can test if templates are working. And we might find new species distributions, who knows? Okay, so that's all nice. And so I'm going to show you some results at last. I know I've been talking for ages. So I'm going to show you the results. 41 templates have been run for vocal mammals. I'm not going to show you all 41. 8 templates for 5 species, sorry, have been run over all 6 sites for all the acoustic data we've got. Okay, so I don't have all the 41 for you because they haven't been run over everything. We've validated a whole bunch of them, and all 8 were successful at detecting species when the species were present. And when we say when the species are present, I mean we saw them there, so we know they're there. So that was kind of good. So I'll just show you some data. So this just shows you all 2 years of data that we have for the whole A2O. And this shows you the number of vocalizations that the thing's getting. This is for Moritan, one of the properties. And it just shows you this is dingoes and this is the hits we get for dingoes. Now, what I want you to see here, you see how there's these gray bars here. The gray bars are the amount of time that we spent at the sites. So Moritan, we managed to go to all the time. And that's how much time, that teensy little itty bit of time. Meanwhile, our recorders are recording away all this time. So you'd think there'd be an opportunity to hear things that you didn't necessarily see. And believe it or not, that does work. So we've got here, these blue ones are all ones that we actually saw and we also heard. But the purple one here, the possums, this is brush-tailed possum, We did not see it, even though the sound hit we got was when we were actually there. Nobody saw a possum. Nobody has seen a possum at the site, but we've heard them there. We know they're there. And so it worked. That was kind of cool. The red ones, however, a bit of a different story. The red ones are ones that we know are there. We actually saw them there, but we don't hear them. And it's interesting because you see that one of those is a cat. Actually, maybe you won't see that. Is that obvious? It's a cat. Anyway, we don't seem to ever hear cats. So there's two possibilities. The fact that we never hear a cat might mean, we never get hits with cats anyway. That might mean that cats never say anything, which is fairly likely. They don't meow very much. But another possibility is that our template's not good enough. So if you really, really wanted to know if there was cats there, you might engineer your template some more and play with it some more and see if you could get, or try different sounds that cats make, things like that. For pigs, we know pigs are there. And again, we didn't get a sound. We didn't get a hit with the pig. So, so far our templates are working moderately well. We are getting hits where we expect to get hits and even some that we don't, but some animals will just never get. And it may be because they don't make any sound. This shows you the same data for another site. This is for Wombiana. The green ones are, again, this is possums, cows, free-tailed bat, dingo, kangaroo, camel, and fruit bats. And those were all things we knew were there, and we heard them. We actually heard them on the recordings. And these are things we also knew were there and did not hear in this. So this is a glider, and this is a cat again. And so there may be things that don't make enough noise for us to ever pick them up, and we're not sure whether that's the case, but we can certainly get a certain number of animals using our templates. So that's good to know. So out of the 8 ones that have been run over everything, we detected 8 out of 10 species. So that's 80% of the species we can do using the templates, which was good. The templates detected 1 out of 10 species with sound only, and that doesn't sound like very many. What I'm hoping is that when they finish with the 41, there will be more hits than we have already on things that we weren't expecting. And some species like cats and gliders either need new templates or we can't hear them. And it could be that you just can't hear them. So I thought that meant acoustics makes a reasonable option instead of visits for the, what do you call it, for the survey, for finding animals. We got 80% of the animals that way, for mammals. Now, Grace has been knocking herself out looking at templates for birds, using templates for birds. And this just shows you diurnal patterns of calling. This is for bubuk owls. So this is diurnal. So this is over the day and it's solar time there. So that's midday, that's midnight. It's calls for owls, calls for doves, peaceful doves, calls for peaceful doves, the world's most boring bird, and calls for magpie larks. And what you can see is that we're getting reasonable diurnal patterns even using these templates. So not only can we get hits and say they're there, we might actually be able to get a little bit more biology out of these things, looking at diurnal patterns of activity. Okay, but then using the templates, we ran into some challenges. So, you just keep running into challenges. So each template has to be run individually. So every single one of these templates has to be run individually over the data. Now, we didn't think that was a big problem. As a matter of fact, we thought that was a real benefit because we could just run them using the HPC, everything would be cool. However, once you get an honors student, a postdoc, and several PhD students all running templates on the HPC doesn't like it one bit and it falls over. So fairly quickly, it depends on the HPC, and actually turns out to be more work than we thought it was, more difficult than we thought it was going to be. Checking the output can take a while, given that there's hundreds and hundreds and hundreds. You saw in my other slide there, where is it? Somewhere I had some information. So for 5, that's 1,056 hits by only checking the top 2 daily hits. And that was just for 8 templates, okay? So that's quite a lot. So you're still checking a lot of data. And recall can be really low. Now that recall is when you miss a thing, when a thing is there and you don't hit it, okay? So recall can be low, which is sometimes not a problem because we don't need to have every call of an animal. We only have to have one call of that animal. So it's not a terrible problem, but it can be a problem if we want to really understand biology of things and use them for this. And then there's these things, bush stone curlews. So bush stone curlews call at night, and these are the template outputs for bush stone curlews. And you can see it's not too bad. It looks like maybe they call at night and in the evening, and then it kind of, in the middle of the night, and then in the morning, and then maybe in the evening and everything. And that's kind of okay. But this is the output for each one of our sites. At our sites, it looks as though they call it dawn here, only at night in Andara, at dawn again and throughout the day at this one, dawn here and in the evening, here only in the evening, and here all throughout the day. And so it's not a very likely pattern for bush stone curlew, and we know that our hits for bush stone curlew are very weak and not very high probability of being correct. And so that's sad. So then I went into the abyss. I was like, oh my God, we had our type templates and that was great and it worked. And now we can't use the templates. They're fine for mammals, but they're going to be hopeless for birds. What are we going to do? And thank goodness, BirdNet got itself together and can now do a lot of Australian birds species. So we can get 137 species just around Townsville using BirdNet. BirdNet is a recognizer. They've built recognizers for every single bird because Cornell Biology Lab has a lot more money than JCU, I guess. But anyway, they've done it. So fantastic. So we can use BirdNet to search for birds. But remember, we're not only doing birds. We can run BirdNet recognizers over the data and find birds. Fantastic. We can also create new recognizers, say for mammals and amphibians, and also for rare birds that don't have things yet, using a few calls and these things called embeddings. Okay, so here we are, transformation again. So we're being transformed by BirdNet. So what are embeddings? No one knows what embeddings are. That's my theory. So embeddings are here. We've got a vector of call features. So here we are back with, remember I told you in the CNN, we needed a vector of call features. Well, here we go. Vector of call features, frequency, length of call, all that kind of stuff. We encode the vector. It goes up. I don't know. They do some machine learning magic-y stuff. And then what happens is they cluster the data. This may sound familiar as well. So they cluster the data and they look at the Euclidean distance between your one call that you have and what they think it is, because they have a lot of recognizers in the data. So they're looking at Euclidean distances, and by figuring out what the Euclidean distance is for a particular call, they can then say, oh, it's really probably close to a bush stone curlew. So we think it's a bush stone curlew. And so then it classifies it into a target class, which is cool. So we can do that. So the coolest thing, so this is cool for birds. So we can just do this because they have a lot of species of birds. But if they don't have a species, we can still do it. And so this is Slade again. So Slade has just created some results from BirdNet embeddings for a frog. So this is for Limnadenastes parani, which is probably called something else now, but used to be Limnadenastes parani. This is the probability of a correct classification, okay, given a particular Euclidean distance from the middle of a cluster. So he's put in a call of a Limnadenastes parani, a few calls of Limnadenastes parani, and now he's looking at the Euclidean distance between, in a cluster, he shoves it into BirdNet, and BirdNet tells him how far away it is from his call, how similar it is to a call. So he then picks a cutoff. We're big on cutoffs in our lab. And so we pick a cutoff where it's very unlikely to be something else. It's very likely to be the correct call. So basically you pick a Euclidean distance that says it's probably going to be that thing, which is not unlike the templates, which is cool. So anyway, we do that. And then you get a graph that looks like this. So this is Limnadenastes parani calling. And this shows you in October, this is at Tarcutta. I don't know if anyone's been down to Tarcutta. It's a beautiful AWC property, I think. Or no, it's Bush Heritage property on the Victorian border. Anyway, calling in summer, like you might expect. Calling in summer and calling in summer. With wetter and drier summers with more and less calls. So, yay, you can do it. So the other thing we can do, which is really cool, is we didn't waste all our time. So now I've been dragged out of the abyss. because we didn't waste all our time with those templates, because the templates give us a whole bunch of hits that are correct and hits that are wrong very easily. We can get 1 million right and wrong hits, high quality and low quality hits, and we can feed those into a CNN, and we can use them to train a CNN, so we can get, and we can use the BirdNet architecture to train CNNs. So we can use correct and incorrect classifications to do that. This reduces the need for citizen science, which is good for Slade. It selects calls that are similar, and it can train CNNs faster. And so you can basically use it to give it the features, and it can learn the features, and it can classify birds. So that's cool. Okay, so here we are. We've been full circle. We started out with CNNs, and I guess in the end, our threshold guardians were correct. We needed CNNs. But now we can build them a lot more quickly and a lot more easily than we could before when we started. This all started probably almost 3 years ago now. Okay, so in conclusion, you have noticed, because you had to go through it, it's been a long journey to get here. We can just about get species lists out of our data. Most species are accessible by some method, but it's actually going to be, Will, to answer your question from the other day, it's going to be a bunch of different methods. So we might use templates for the mammals for some complicated calls that are very long. We might use CNNs for some things. We probably won't build CNNs for everything. We will use BirdNet's already existing things for other things. So it's going to be different strokes for different folks. It's not such a big surprise, really. Acoustics is quite a good way to inventory species, I think. I don't think I did a really fantastic job convincing you of that, but it is possible after you spend 2 years tearing your hair out. Technology is changing really, really fast. So probably by the time we finish, there'll be CNNs for everything anyway. And now we're going to do threatened species. So we've moved on now in my lab, so that we are working on, we feel like we have enough techniques to get the common species that we can easily get, and we're moving on to threatened species. And so this actually does link back to my very first slide, because my very first slide had a swift parrot on it. And the swift parrot is one of the things that we have seen and heard on, we've heard it in the data. So the swift parrot is an example of a threatened species, also koalas, that we hear on the data that we can get with a variety of different techniques that will allow us to inventory probably the important things. Although I do think that this is still a useful method, because we need to know these things are still useful to do, even on common species, because we don't always notice, the passenger pigeon, for example, we don't always notice very common species declining. And so hopefully we can notice them using these kinds of recording, which is why we got into it in the first place. That's it.